Good morning. Right. Well, thank you. Nice to see such a full lecture theatre. If there are any questions as we go along, just shout up, because we might as well do that, or we can wait till the end, or there'll be no questions. We'll soon see. So thank you, Stephen, for inviting me to come back to give a talk on biophysics in drug discovery. My name's Chris, and I'm a principal scientist at AstraZeneca in the biophysics team, working across the road at the DISC. I've been at AstraZeneca for just over 10 years now in the biophysics team. My main focus is non-NMR biophysics. So if you ask me any NMR questions, I probably won't be able to help you unless they're really easy. But if it's anything else, I should be good to go. Okay, so this presentation is in two parts. The first part is very short, it's an introduction. It's just briefly introducing what is drug discovery and where does biophysics fit into that process. Then I'll give a number of examples of project impact where biophysics has provided information that has been important for projects. I'll be covering things like reagent characterization, hit finding, looking for things that bind to the targets, orthogonal validation, so confirming hits that have come from other techniques. And then the last section is a bit more general, and it's hit characterization. What can we learn about the compounds? Drug discovery is a long process. On average, in the past, it's taken in excess of 10 years to get from the beginning of a project to a marketed medicine. And that is if things actually get that far. There's quite a lot of attrition during the process for different reasons. We can think about the process as discrete steps. The first thing we need to do is identify a target. So this is a biomolecule that's involved in the disease. Importantly, it's one that we need to have confidence that if we find something that binds to it, we will be able to modulate its activity or potentially degrade it, or in some other way cause it to stop promoting the disease state. Once we have chosen the target and we're confident that it's a good target for a project, we initiate the hit identification stage of the process. In this process, we are screening for initial compounds that we call hits that we find interact with the target. This could be done by a number of methods. People are most familiar with things like high throughput screening, which can be done biochemically or in a cellular context. But there are other hit finding strategies, such as affinity screening or DNA encoded library screening or fragment screening, which I will touch on in a few slides. Also, during this part of the process, we need to confirm the hits are real using orthogonal technologies and assays. This is in order to build confidence that we will be following up something which really does bind to the target. There's nothing worse than you run a screen, you don't validate the hits, and quite a long way down the process, you realize actually it was an artifact of the assay and it doesn't really interact with the target in the way you want. During this part of the process, we will do a limited amount, maybe a bit more than limited, depending on the technique, of near neighbor testing. So choosing compounds that we have available in our collection or that we can purchase easily to quickly explore the hit space to find slightly better molecules that bind to the target. Once we've found a couple of hit series, the next step is hit lead or lead identification. This is where there will be some medicinal chemistry done and characterization of the compounds, where we want to develop them into leads, which are compounds with higher potency than hits. Here we're looking to increase the potency and potentially improve their properties, such as solubility, lipophilicity, things like that. And if relevant for the project, we might start looking at selectivity at this point too. Once we have found or developed our hits into leads, I should say, the next stage is lead optimization. During this process, there is significant medicinal chemistry investment. And I mean investment in terms of time, but also money. So we need to be confident that the leads that we've identified are good enough. During this part of the process, this is where we refine the lead series, improve their potency, really focusing on potency, selectivity, the pharmacokinetics and dynamics, safety profiles, things like that, to get to the best point we can and identify a molecule we think could be a good clinical candidate. After that, we've got preclinical development. This part really focuses on building the data package that's needed for regulatory filings and things like that, that allow us then to go into clinical trials. And then the final stage before stages, I guess, because it is split into multiple steps, but I'm not showing them here, are the clinical trials. This is where the molecules make their way, or we put them into humans and assess their safety, efficacy, and actually also, if we're not the first company with a drug in this space, is it the best? And then finally, we have a marketed medicine, which we can give to patients. And that is why we go to work every day, to try to get to that point. In terms of where biophysics plays a role, it really is in the early stages of the process, the ones I've highlighted in this greeny blue color. I've pulled out a few examples of areas that biophysics can contribute. They're mainly on the slide because they're in the deck. So there are other things that we can contribute to in projects. In terms of the earliest activities, once we've selected the target and it's been validated, if we're doing any sort of in vitro hit finding, we need to have reagents in order to do that. So recombinant proteins, if the protein is a target. Where biophysics can contribute here is QC of the protein. And by that, I mean, is the protein folded? If we have known interactors, does it bind to those things? Another step, another part of this could be evaluating tool compounds. Whenever we do a scoping for a project, we look at the literature and see whether there are reported ligands for the targets. Unfortunately, I would say in the majority of cases, the things that we are finding in the literature that are reported to bind do not. So, we need to make sure that we have validated tool compounds. And actually, this is where biophysics is important, because these compounds might be active in a biochemical assay, but behaving artifactually. But we do direct binding experiments, and we either don't see any binding, or maybe we see that they are completely insoluble or aggregated and things like that. Also, during this part of the process, people will start to think about their assay development. So developing the NMR and SPR assays, the mass spec assays, to help kind of get a head start for the rest of the process. During hit identification, biophysics can have a role in primary screening, so finding the initial hits, and that would be done using fragment-based screening, which I'll talk about later. But I would say the biggest, the largest amount of work that we do is in orthogonal validation of hits that have come from other hit-finding strategies. Because we are truly orthogonal to a lot of those, we've got a fluorescence-based threat assay, for example. When we use SPR, they are truly orthogonal. And then the rest of the work that I've highlighted really can fit into any of the other three categories, I guess. So we've got mechanistic characterization. If we don't have a structure for some reason, can we get information about where compounds bind using some sort of structural biophysics? What are the kinetics of the binding? What are the requirements of the binding? Do you need to have a cofactor present? And how, if it's not there, how does that affect the affinity of things? And then more generally, we'll have compound profiling. So testing compounds regularly for their affinity for the target. And sometimes it's driving the project, other times it's just spot checking the other more high throughput biochemical assays. In terms of the biophysical techniques we have at AstraZeneca, this is much nicer when it's dark. We have quite a few different techniques. I would say the workhorses for us are SPR, NMR, and MS, mass spec. But we do have access to other techniques. And actually, the projects are becoming more and more complex as the easier things have started to be done already. So we recently are using mass spectrometry a lot more to characterize multi-protein complexes. The feeder one that we have, that's also proven useful in projects, and that's used for looking at the hydrodynamic radius of molecules. And then we've got the old faithful ITC, isothermal titration calorimetry, which I'll mention briefly later. We don't use that so much because it needs a lot of protein. If you've done it, it uses a lot of protein, but it is a very good technique, especially when you're trying to problem solve other biophysical techniques. And really that is the strength of having so many techniques at our disposal, because they all say different things about the protein, and that's what I've shown in blue here. These are characteristics of the protein we can determine using these techniques. So they're complementary to each other. If one doesn't work, hopefully another one will. And if they both work, hopefully they give the same answer. And obviously, we can also use biophysics to look at the protein interacting with the ligands. Does it bind? How well does it bind? What's the affinity? What are the kinetics? And we can get some idea of binding site. We use things like NMR and mass spectrometry. Okay, so that's the introduction out of the way. Now I'll move on to the different examples. These first three are, broadly speaking, looking at reagent characterization. Native mass spectrometry is a particular type of mass spectrometry where we infuse the sample into the mass spectrometer in such a way that we aim to maintain the non-covalent interactions that are present, such that when we infuse it into the mass spectrometer and it goes from the solution phase to the gas phase, it maintains the same structure. This is not guaranteed, but it does work. A limitation of the technique is that it's not compatible with non-volatile buffer components. So if you try to infuse it in 20 millimolar heaps with sodium chloride, it's not going to work. You're just wasting your time and making the instrument dirty. So what we need to do is buffer exchange the protein into a volatile buffer. So typically we have ammonium acetate, which is not strictly a buffer, but it does provide some ionic strength to the solution to help keep the protein happy. We also use soft ionization techniques such as nanoelectrospray, really just to maximize the chance that we can infuse it and maintain non-covalent interactions. In doing that, it will allow us to characterize the protein, its ligameric state, and/or ligand binding. And actually, we have had one project where the protein co-purified with a cofactor, and we wouldn't easily have been able to determine that if we hadn't used native mass spec. And that particular project was looking at finding compounds that were competitive with this cofactor. So having it present in the mix is not a great thing. So protein science were then able to optimize the purification to remove the cofactor. We could confirm that using the native mass spectrometry. At the bottom of the slide here is just an example of a native mass spectrum. So on the x-axis, we've got the mass to charge ratio. On the y-axis, which is hidden, but the scale is just the relative intensity. And we can see, hopefully you can see, there are three clusters. I'm not going to do that again because I'm moving a bit, of ions. And if you deconvolute it, they correspond to a monomeric species, a dimeric species, and a tetrameric species. And that is what was reported for this protein. So that was good to see. And it also matched the size exclusion trace, which was quite heterogeneous, but bumpy. And because it was reported and it still binds things, we also verified that using other techniques. We were happy with this protein. Another mass spectrometry technique is hydrogen deuterium exchange mass spectrometry, which hopefully you've heard about in some of the other lectures, the other lecture on mass spec. This is a structural mass spec technique that measures the incorporation of deuterium into a protein's backbone as a function of time. We do this by exposing the protein to a buffer that has an excess of D2O over H2O for different lengths of time. And this little diagram here shows, there's a backbone proton there, and just happens that this one exchanged deuterium. And that's what we're looking for in the mass spec experiment. The key here is that if residues are solvent exposed or in flexible regions, they will exchange quickly or more quickly than residues that are buried or those that are involved in stable secondary structures, because either they're completely occluded from the solvent because they're in the core of the protein, or in the case of secondary structure, you've got hydrogen bonding, intramolecular hydrogen bonding in the structure, which kind of protects the protons from exchanging with the deuterium. When we do the experiment, generally we will digest the protein using pepsin, and that allows us to get peptide level information on where the deuterium has been incorporated. where we've used this in a project, there was a question, and the question was, where are the structured regions? This target was known to have the DNA binding domain, DBD, but the C-terminus was thought to be completely disordered, with the exception of a small region that was reported to have a helical propensity there. So we did an HDX experiment to see if we could get any direct evidence of that. And that's the data shown on the slide. So on this plot, the y-axis is the deuterium uptake. So 0 is it's not taken up any deuterium, and 100% is it's fully exchanged. Each of the different colors are different exposure times, so incubations with deuterium, 0.3 seconds, which is actually 3 seconds, but cold, is the shortest time. What you can see on the left-hand side is the DNA binding domain, and we see different levels of uptake at the different time points, and that is characteristic of something that is folded, and that's what we would expect to see. But on the right-hand side, we can see that all of the lines essentially overlay with each other, which is consistent with a disordered protein, not something with a stable secondary structure. So we don't think this protein does have that helix there, and that was a big question for the project. The last example is isothermal titration calorimetry. ITC, it's a technique that measures heat. And in the context of molecular interactions, we're measuring heat changes that happen upon ligand binding. And we use that to determine thermodynamic parameters. The example I'm going to show is for a tool peptide that was reported that we were going to use as a tool compound in our assays. We wanted to validate that it bounds to the protein target. So to do this, we have a 30 micromolar protein solution that's in the cell of the ITC instrument, into which we titrate aliquots of the peptide. In this case, it was at 500 micromolar. And as the aliquots are injected, we see this decrease in the power that was applied to the reference cell in the instrument, and that indicates that we have a binding event. And as you see, as the injections progress, they become less negative over time, and that's consistent with the fact that as you're injecting it, it's binding, we're saturating the binding sites on the protein. So this looks good qualitatively. In terms of analyzing the data, you just integrate the area within the peaks, and that gives us the delta H versus molar ratio plot here, and we can fit that to a one-site binding model and derive the affinity and the enthalpy. We expected the affinity to be in the hundreds of nanomolar, and that's what we saw, and the N value, the stoichiometry, was 1. We expected it to be 1-to-1 binding, so this validates that the protein is fully ligand binding competent. That's making the assumption that all the concentrations are correct. Okay, so this next section is about its identification, specifically where we can use biophysics to do primary screening. I would say the only type of primary screening, I could be wrong, that we would do in biophysics is fragment screening. What do I mean by that? In the context of this, fragments are small molecules that have a molecular weight of less than 300 Daltons. Because they're small, the amount of chemical space they occupy is also smaller because there are fewer atoms. This means that we can explore that chemical space with much smaller libraries. So a fragment library size is low thousands. Our current fragment library is just under 3,000 compounds for the primary screening. If you compare that to an HTS, high throughput screening library, they are in hundreds of thousands or millions of compounds, so significantly lower. On the flip side of that, because they are small and have low molecular complexity, we don't expect them to have high affinity. That's not to say you can't get really potent fragments, because you can, but generally speaking, they are tens to hundreds of micromolar, even millimolar potencies. Because of this, we need techniques that are sensitive and robust to high concentration screening in order to identify them. And this is where biophysics fits in really well. The 3 techniques that we would use in AstraZeneca to do fragment screening generally are SPR, NMR, and X-ray fragment screening. I'm going to show you some data from an SPR fragment screen, but before I do that, I just thought it would make sense to just briefly recap what I mean by SPR. So SPR stands for surface plasmon resonance, and that's the biophysical principle behind the detection method. But when we talk about SPR, we're really talking about this biocore system from Cytiva. That's the particular flavor that we have at AstraZeneca. One of the key components of the SPR instrument is actually the sensor chip, and because SPR is a biosensor technique. The sensor chip itself is a glass cover slip that's coated with a bit of metal and then specifically a gold layer on top, and that's what's responsible for the surface plasmon resonance. We immobilize our target molecules onto sensor chips, and it actually, in order to immobilize more target, because the signal we see in our experiment is proportional to how much you immobilize, there is a thin 100 nanometer layer of carboxymethyl dextran on there, where we can actually attach our macromolecules to the surface. As I just said, the SPR response is proportional to the mass that's absorbed at the surface of the sensor chip. So when we immobilize the protein, the signal changes, but also if things bind to the protein on the surface, that will also change the signal because of the increased mass. In terms of how you mobilize the protein, the very first way it was done was using covalent cross-linking. There are 3 carboxylic acids in the matrix that you can activate using EDC and HS, and then you would inject your protein across the surface. When you do this, actually you do it at a pH below the PI, such that the protein is positively charged and attracted to the negatively charged surface. In doing that, quite often, it means that when you immobilize the protein, it's no longer fully functional, either because it really does not like low pH, or because you've introduced so many cross-links between lysines on the protein and the surface. So the 2 ways that we most commonly use now for immobilizing proteins are using a nickel NTA functionalized sensor chip and having a his-tagged target, so that the his-tag target is attracted to the surface by virtue of the affinity between the nickel NTA and the his-tag. If you do that, then typically you would do a small amount of cross-linking as well, just because the affinity of a mono his-tag is not very high, so it drifts off the surface quite quickly. But by and large, the best way of doing it, or the favorite way of doing it, it's my favorite way of doing it, is using a streptavidin functional-like sensor chip and having a biotin-related target. And the best way is having an Abi-tagged protein, because you know that you will only have one biotin on there and you know where it is. So that is the best way of doing it, I think. The other really important component of an SPR instrument is the microfluidics. And I took a photo of a IFC, integrated microfluidic cartridge, from an SPR instrument that came out during a PM. That's on the slide there. You can see all these little divots within there. I mean, this is only about that big. This is where your sample flow goes up and stuff. But right in the center there is this protrusion, which forms flow cells in the instrument. And what happens is when you put your sensor chip into the instrument, the IFC docks onto the sensor chip. And where you've got these divots in the center, they form the flow cells in which you can inject things across the surface of your sensor chip. For a T200, which I think you have here, this would be the flow cell layout. So we have 4 flow cells that are all connected. You can address them individually, but generally you would inject things across all of them unless you were immobilizing. You would always have one flow cell that is left alone. So you would treat it in exactly the same way as you have all the other flow cells that you've immobilized something, except you don't mobilize anything. And this allows it to be a control that you can subtract from the other flow cells to correct for systematic artifacts in the data. So here you would have 3 opportunities to mobilize other targets. So you've got your immobilized target on the sensor chip, it's in the system. How do we actually test that things bind? If you want to determine the affinity or kinetics of something, you would inject a dose response titration series. And that's what this little animation shows here. So initially there's a baseline where it's just buffer flowing across the surface. And then at 0 seconds, because that's where I've aligned the sensorgrams, the flow switches and the analyte is injected. At 60 seconds, the flow is stopped. Well, it's not swapped, it's switched so that buffer is then flowed across the surface. So any analyte in the flow system is flushed out, and also any analyte that is bound and then just has dissociated from the immobilized target is also washed away. And then we get these classic SPR sensograms. This is real data. I was going to simulate it, but I thought I'd give you some real data. And once we have this data, then we can fit it. If it looks like this, and you have clear binding kinetics, so curvature in the sensograms, then you would use kinetic fitting. And this involves just using the software which has differential equations which define these equilibria, and it fits them using nonlinear least squares regression. And from that, you would get the K on and the K off, and obviously then the KD for the interaction. If you haven't got any kinetics because it's too weak and too fast, or too fast to resolve the kinetics, you would do what we call steady state fitting. And that's where you would take the response at steady state, where you've got a plateau in the sensograms. Most of these have just about reached that. So you would take it about this point here, and you'll plot those versus the concentration and fit it to that equation there to get the KD. Anyway, back to fragment screening. We've immobilized our target. So now we need to screen the library. Because SPR is not super high throughput, we screen at a single concentration, even though it's not that many compounds to test or many fragments to test. The concentration of screening is chosen as a compromise based on the fact that we are expecting to see weak things. So we need to use a concentration high enough to actually get binding, but also we want to minimize the risk of poor behaviors, insolubility, non-specific binding. And so 300 micromolar is a reasonable number there. So the 2,700 odd fragments were tested against this target, and we found 94 compounds that were active. And by active, I mean they didn't show any poor behaviors, and they were significantly different from the median response for the rest. There were 74 compounds we flagged as irregular, so they had something strange about them, and the rest were inactive. And you can see on the plot on the right, each of the points there is a different fragment, and on the y-axis, it's the response they gave at the beginning of the injection. So with the primary hit list there, we want to find the fragments that are most promising. Pulled out one fragment here, fragment 1, because it shows what we expect to see in terms of shape for a sensogram of a fragment. Because fragments should be weak, or most likely to be weak, we don't really expect to see any kinetics to their binding, because unless they have an incredibly slow on rate, they will have to have a very fast off rate. So it should look like this kind of top hat square pulse type sensogram. This is what we expect to see. Once we've got our actives that we think are worth following up, we then do the standard thing of doing a dose response to try to determine the affinity of the fragments. So that's what was done with this fragment here. It was titrated from 500 micromolar, and we found it had a KD of 330 micromolar. So not the best, but not horrendous. Definitely worth following up. The good thing about the SPR instrument is that as well as getting a response number, we also have the sensograms, and that allows us to identify irregular behaviors. I've pulled out 2 examples of irregular, poorly behaved compounds here. The sensograms are shown on the right, left, sorry. The first thing to note is that the responses are very high. So the y-axis is the response as a percent of what we would expect to see if you'd saturated the surface with the compound. So the top one, it's got to above 600%, massively high, super stoichiometric, not one-to-one binding. And the one underneath, it's not even started to plateau. So these are not things worth looking at. Secondary to that, we've got multiphasic association. Binding should be single exponential unless you've set your assay up really badly. So this is not the desired behavior. And the same for dissociation. In this case, they're both slow and multiphasic, so not worth following up. The next section is about orthogonal validation. It still sits within hit identification, but now we're looking at hits that have come from other screening technologies. One such screening technology or paradigm is affinity screening, and this is just looking for things that bind to your target, not necessarily affect the function in any way, but just bind to it. We do affinity screening at AZ, and it's high throughput. But by virtue of being high throughput and looking for things that bind, the false positive rate is going to be high because we are looking for things that bind. Things that bind non-specifically will bind, so they will become be flagged as actives. So biophysics has a key role here in validating hits from these sorts of screens to find genuine hits that have measurable affinity for the targets. So the target that I'm going to talk about is an unprecedented transcription factor. By unprecedented, I mean in particular for the domain of it that we were looking at, there were no reported tools and no known binders for it. So we didn't really have anything to validate our assays. This is where affinity screening is very powerful because you don't necessarily need to have tools in order to do the screening, and actually you can find tools by doing it. So this work was done in the hit discovery department where they screened this target using two methods: affinity selection mass spectrometry, which looks at screening pools of compounds for binding to the target, and they screened 200,000 compounds, and they used differential scanning fluorimetry, DSF, or thermal shift. They screened just under 100,000 compounds using that and looking at the stabilization of the melting temperature of the protein. So once they had the hit list, they triaged it, looked at it for chemical structures they will be interested in working on, looking at properties, purity, those sorts of things. And at this point is when we would bring in the biophysics. The challenge for this project was the fact that this is an unprecedented target. So to build an SPR assay, for example, we really do need a tool, because how will you validate the protein is folded and active on the surface if you don't have one? So that was a challenge. So what we did was we asked the chemists to prioritize the hits, and we got 43 hits given to us. Fortunately for this project, the domain of interest was quite small. It was about 25 kilodaltons and expressed really well, which meant that we could make labeled protein for 2D NMR. And that was actually why 43 hits were chosen, to give a manageable number of things to look at. So that's what we did. The 43 hits were tested by 2D NMR, looking for chemical shift perturbations, CSPs. They were tested at a single concentration of 200 micromolar. And an example of the data that was collected in that one of those experiments is shown here. So you've got the 2D NMR spectrum, the proton chemical shift on the x-axis, the nitrogen on the y, and these are the cross peaks in the middle. In blue, we see the apoprotein resonances, and in red, we see the compound, or the resonances in the presence of the compound. The fact that all the peaks don't sit on top of each other tells us that there is a binding interaction here. From those 43 hits, 3 of them showed behaviors like the one on the slide, so indicated binding. So as with the SPR example, the next step is doing a titration to try and work out how potently they bind. And they were weak, but measurable for 2 of the compounds. So it was promising, because we could then indicates these are showing specific interactions. The chemists then went back and looked at the AZ collection and identified some near neighbors, so molecules that look similar to the hits. And these were then tested by NMR again, initially a single point, and then in dose response. And that led to the identification of compound 4, which had a KD of less than 20 micromolar. So excellent, really. This is a great starting point, and a very useful tool, because with this validated compound in hand, we were then able to develop an SPR assay. So that's what we did. On the left, you can see the SPR sensegrams for that less than 20 micromolar compound identified in the NMR experiment. On the right of that figure there, we've got the steady state responses plotted versus concentration. And if you fit that to the equation on the slide, we get the KD, which came out as 6.3 micromolar, and the Rmax. So at the top of the slide, you can see another equation just for reference, but we're able to work out what we expect to see for the response level based on the molecular weight of the compound relative to the molecular weight of what was immobilized and how much was immobilized. And it's about 130%, which is higher than 100%, but it's within the error, and that's fine. So it shows that the protein in this optimized assay is nice and happy. With this assay in place, it allowed the project access to a higher throughput assay that was needed to characterize new molecules as they were designed to improve the affinity. The project is now at the stage where we've got low nanomolar compounds, and it is still being driven by the SPR assay. So it's a really good result. Also, in this project, we applied some structural biophysics. The X-ray crystal structure was elusive, wasn't crystallizing. So we used some structural solution phase structural biology techniques to try to understand where the compounds might be binding. The first example is 2D NMR again. When we did the screen, we didn't have the assignment, but as the project progressed, Mark managed to get the assignment for the 2D NMR spectrum, so we could see which residues were perturbed when the compound was binding to the protein. And we also did an HDXMS experiment, which gave very similar results, showing that regions of the N-terminus of the protein were perturbed when the compound was present. And this data was used to help guide docking efforts to understand where the compound and how the compound might be binding. The last section is a bit more general in its mechanistic characterization. So the aim of any project, well, the biggest aim is to find molecules that bind really potently to the target. But actually, at a certain point, that becomes problematic, because any solution phase assays will reach what is called the tight binding limit, or a potency limit, where although you're making changes to the molecules, they just don't seem to be making any improvements. And that's exactly what happened in this project. So on the histogram here, these are all the compounds that were tested in a particular biochemical assay. And you can see that they're all starting to cluster around a PIC50 of 8, which is the same as an IC50 of 10 nanomolar. And actually, the enzyme concentration in this assay was 20 nanomolar, sorry, my goodness. So we are at the tight binding limit here. So actually, the chemists from this data alone have no idea whether the changes they're making are improving anything. Fortunately, SPR does not have that limitation because you're in flow. It's a flow-based system, so you don't get ligand depletion. You're constantly replenishing the surface with new compound. And what we can see here from this histogram is that there are a number of compounds that show significantly higher potency than 10 nanomolar, down to about 2 picomolar. So this tells us that we can improve on the chemistry, even though the biochemical assay said otherwise. This plot here will show the correlation between the biochemical assay results and the SPR assay results. So if we focus on the top right-hand corner of the plot, there's a good correlation between the two assays. But when we get to this tight binding limit, you can see that the correlation is no longer there. But we do then see this spread of KDs, PKDs. So we are improving the potency, but also, I think, importantly, we are still inhibiting the protein, because binding does not necessarily mean inhibition. But in this case, we're all good. And actually, this project was also driven using a cell-based assay, which correlated really well with the SPR. And again, it didn't have this same tight binding limitation. And my last example is about molecular glues, and specifically using the feeder to look at those. By molecular glues, I mean monovalent small molecules to induce or enhance existing complexes forming between two proteins. As I said, we can use feeder or flow-induced dispersion analysis to characterize these interactions and their targets. How do we do this? Well, feeder is a technique that we can use to determine the hydrodynamic radius of molecules. So if things are in a complex, they will get bigger, that will change, we can measure that. This is a picture of the instrument we have in the lab. On the right-hand side, you've got the light source, and the top of the instrument is where there's a very, very long capillary, very thin capillary, which you push your samples through under conditions that you have tailored dispersion. That allows us to do the feeder measurement. We've got a really crude animation here to kind of demonstrate the principle. The instrument actually only runs one sample at a time, but I'm going to run these in parallel so you can see the difference between the two. So the first thing that the instrument does is it takes up a sample of your, a small amount of your sample. In the top capillary, we've got dye only, and in the bottom capillary, we've got a protein that has been labeled with a dye. So we typically use Alexa 488 or 647 or fluorescein or something like that. So it takes up a small amount of sample, then pushes it into the capillary. I think it's 40 nanoliters that it takes. And then that sample is then pushed through the capillary. And because we have such a thin capillary and the flow rates are set appropriately, we have laminar flow in the system. That causes there to be a parabolic flow profile. So as it gets pushed through the capillary, you kind of get this dispersion effect. In the case of the labeled protein, it's a much bigger molecule, so we have a slower diffusion, so lower diffusion coefficient. And actually the important diffusion here is this radial diffusion. And what that does is if it was higher, it would stop this spreading from happening. So because it's slower, you get this more diffusion, more dispersion. Whereas for the dye only, because it is, it's diffusing between the outside and inside of the capillary, you kind of counteract this spreading effect. So that goes past the detector, and you get this tailogram. It looks like this. So for things that are smaller, you see a narrower, sharper tailogram, and things that are bigger with a larger hydrodynamic radius, they are broader and flatter. And the software will then fit this peak to a Gaussian and uses that to determine the hydrodynamic radius of the molecules that it detected. In terms of how we use that in a project, there was a project that was looking for molecular glues that will glue a peptide to an E3 ligase. So we obtained the peptide with a fluorescein attached to it. When using feeder, it's important to label the smaller thing, because when something small becomes bigger, you get a much bigger assay window. So to characterize this interaction, the peptide was kept at 10 nanomolar, and the protein was titrated. On the y-axis here, you can see the measured hydrodynamic radius of the labeled peptide in the presence of different concentrations of the ligase. You can fit that to an equation, and you get 46 nanomolar as the affinity. In this project, there was a reported molecular glue that we were able to validate. And so you repeat the same experiment, but in the presence of 20 micromolar, in this case, molecular glue. And what we see is the dose response curve shifts to the left, indicating a higher potency. And when you fit it, the KD comes out at 7 nanomolar. So the KD for the E3 ligase, the peptide in the presence of the glue, is significantly higher. This assay was then used to ultimately devalidate hits from a TR-FRET-based molecular glue screen. Although it's not completely orthogonal, because they both use fluorescence, they gave useful data. So that was a good use of this particular technology. And then the last slide, I can't get off it, is just a summary and the acknowledgements. So hopefully I've convinced you that we use biophysics to add value across multiple stages of the drug discovery process. Gave you a few examples in QC and screening, orthogonal validation, mechanistic characterization. Different biophysical techniques have unique strengths and weaknesses, and the fact that we have lots available to us means that we can answer questions comprehensively. And ultimately, combining the biophysical data with the biochemical and cellular data allows us to build confidence in hits and compounds, and understand the mechanism and push the projects forward. Lastly, just need to acknowledge the team, the biophysics team. Everyone there works across the road at the disk. They contributed data to the slides. And thank you for listening.